So we said transcription happens in the nucleus, and we go from DNA to a section of pre-mRNA that's edited, so the introns are taken out, the exons are put back together, that's called splicing, and you get a 5' cap and then a poly A tail. That mRNA then leaves the nucleus, again M stands for messenger, and then it's used in the process of translation. So that's where we actually build a polypeptide or build a protein. So this is where ribosomes are going to be involved. There's a couple of other types of RNA that are also involved, so we'll talk about those. First one is tRNA. The T stands for transfer. So tRNA is involved in the process of translation. It looks like this. So this is a simplified version of tRNA. This is what you usually see, bless you, if you look at animations or videos, because this is less complicated than this structure or this structure. But the tRNA is a strand of RNA that folds on itself, and it has an end that an amino acid can attach to, and then it has an end that has an anticodon. So the anticodon is complementary to the codon. Do you remember where the codon is? What molecule has the codon? The mRNA. So the tRNA has a sequence called an anticodon that's like the opposite of that, so that it brings the right amino acid for the right section of the mRNA. So the tRNA will only bind to one type of amino acid. There are enzymes that help with that process. So there's a different enzyme for every single type of amino acid and every type of transfer RNA to attach them together. But how many amino acids are there? 20. So we got 20 different kinds of enzymes. So that's a lot for your body to have to handle and figure out, but it makes it work. The tRNA then takes the amino acid that's been attached to it up here to the ribosome, and it's added to the protein in the correct order. Okay, so the body, as it's making proteins, is constantly attaching new amino acids that are free floating to tRNA molecules to take those to the ribosomes for translation. Okay, that's kind of the gist of what's on this page. You can read a little bit more about it, but the purpose of an anticodon is to make sure that you have the correct amino acid brought to the ribosome at the right time so that the protein can be made in the right order. Okay, so we've talked about the fact that ribosomes build proteins. We usually use the term polypeptide because the chain that you get of amino acids after the ribosome is done might not be the final protein. Sometimes that gets edited a little bit, or you have to take two different chains of amino acids and have them interact together to make your final protein. Okay, so usually we say polypeptides instead of amino acids. The ribosome, what's that?
Polypeptide, amino acids, same thing.
Sorry, usually we say polypeptide instead of protein,
Okay.
but they're usually interchangeable. So if I ask a question about polypeptides, I'm asking about proteins. Okay. The ribosome's made of rRNA. R stands for ribosomal. So M is for messenger, T is for transfer, R is for ribosomal. And the ribosome's two parts. So there's a bigger part on top, it's called the large subunit. The smaller part on top is the small subunit. And one of the interesting things is there's a large and a small subunit in both prokaryotes and eukaryotes. But in eukaryotes, the sequence is different, so it's a slightly bigger ribosome, and it's different enough that it's medically important. The reason it's medically important, this is described here underneath where my sticky note is that scientists, doctors, people developing medicines can use the differences to target just prokaryotes and not affect your cells or eukaryotic cells. So if we can stop the translation of proteins in bacteria, which are prokaryotes, we can kill bacteria and it won't affect your cells at all. That's how some antibiotics work, to stop bacteria from reproducing. So your cells aren't harmed, but all the bacteria cells can't make proteins, so they die after a little bit of time. Okay, so it's kind of cool. We can use small differences like that to make our medicines more effective. Something you'll need to know, and we'll talk about this as we talk about the process of translation, are these A, P, and E sites. So when you're looking at the ribosome, the A site, then the P site, and the E site is right here. so it's not labeled in most of these diagrams. But when you're looking at the process of translation, the codons start here and then they move to the left. So you'll have a start codon and then it moves to the left. So as the mRNA is moving through, it's moving from one site to the next in sections of 3. Remember that triplet code is how you get your codons, and then the anticodons there in sets of 3. Okay. So the good thing and then potentially confusing thing, transcription had 3 steps: initiation, elongation, and termination. We use the same terms to talk about translation. So it's the same names, but the process is different because they're 2 different processes overall. So same terms, but what happens in them is different. Okay. Initiation is still talking about starting the process of transcription. So in order for that to happen, we need the start codon. Do you remember what the start codon sequence is? AUG. What amino acid does it code for? M something. That's a good start. Methionine, MET. Okay, is usually how it's abbreviated. So wherever that AUG sequence is, that's where the mRNA is going to start in the ribosome in order to translate the mRNA into a protein. Okay, so that initiation codon is important. And that's what I have on my notes on this side. So that initiation codon is AUG. Again, translation has the same 3 steps as transcription. They're just, the process is different. So the start codon starts in the, and I wrote A site, but that's not right. The start codon actually starts in the P site. It starts in the middle of the 3 different places where codons are being used inside the ribosome, because it starts like this. So there's a small subunit and the large subunit. They're not connected initially, so they're completely separated from each other. So you have a ribosome that's like open for business. It's not connected to each other. And then an mRNA molecule slides into place. So that start codon slides into the middle slot, and the large subunit clamps on top of that. Once that happens, initiation is done. So the process of initiation is the mRNA lining up in the right place, and then the subunit pieces connecting together so the ribosome can start sticking amino acids together. Okay, so you said the start sequence is AUG, the start codon, so that the amino acid that's attached to that is methionine. This is now the first amino acid in the protein. So in most of your cells, as the polypeptides are being made, the first amino acid that comes out of this tunnel as the polypeptide's growing is methionine. Okay. Next step, elongation. So now we're adding more amino acids to this. So the next transfer RNA molecule has to slide in place here. I'm going to move this. So we're looking at this step here, this loop. So this is the next transfer RNA. It comes into the A site. You can think of A as arriving. So it's where the transfer RNA brings the amino acid to. Okay. So this is codon recognition. You have to get the right transfer RNA in this place so that it matches up the right way, which means you have the right amino acid. If the codon's not recognized the right way, then you'd be bringing the wrong amino acid to the chain, so that would be a problem. So codon recognition is the first part of elongation. The second part is peptide bond formation. So let's see if you remember from macromolecules. What are peptide bonds formed between? That's a sense that I'm getting. So let me ask a different question. I just talked about polypeptides and proteins. So what makes a protein? What are the building blocks of proteins? Amino acids. What are the building blocks of polypeptides? Amino acids. So the link between amino acids is the peptide bond. Okay. So after you have your methionine, that's the start, the very first amino acid, when another amino acid is brought into place after the codon recognition, then they have to be linked together. So that's what happens after you get the transfer RNA in place. You get a bond between the 2 amino acids. Okay, so now we have a new bond. And then translocation happens. So what's translocation mean? Give me a guess. Moving. Like, yeah, literally moving. So the whole strand of mRNA slides over. which means all the transfer RNAs move over one. And now the A site is open again. You have the transfer RNA that you just had brought in the P site, and from the E site, you can think of E as exit. So the tRNA that was there no longer has an amino acid, it leaves the ribosome. Okay? So the A site is where tRNA come in, the E site is where they leave, the P site is in the middle, kind of as it's hanging out, waiting for another tRNA to bring another amino acid. Okay? So we've got codon recognition, peptide formation, and then translocation. So you should be able to talk about the sites that are involved and kind of describe how mRNA moves between those three, how the tRNA moves between those three sites. Does that make sense? Okay, so this elongation process happens over and over and over and over until we get to the stop codon. So remember, there's only one start codon, one sequence, but there were 3 different stop codons. Once the ribosome reaches the stop codon, it literally just goes, okay, I'm done, and the subunits pull apart, and then that's it. That's the end of translation. Okay, that's mentioned down here. My notes on it are on this post-it note here, which is just literally termination. The stop codon signals the ribosome to unhook, so then it stops translating. Okay, this section is pretty much a review of everything from 10.6 to now. So it goes over and talks about big picture transcription and translation. What are the steps? What do they look like? So if you're looking for, I don't quite think I understand what's going on, this is a good section to go back and look at. This would be a good section to review in your notes or specifically in the book right before a quiz or maybe right before the test to help you review terms and where things happen. It's faster than reading the whole book again or maybe even reading your notes, depending on how lengthy your notes are. Okay? Questions so far about translation? Not so much? Okay, so last section I'm talking about talks about mutations. What's a mutation? Okay, so sure. What caused the Teenage Mutant Ninja Turtles to be different than normal turtles?
There's like some goo. Yeah.
Okay, so we would call that a chemical mutagen, right?
Yeah, it's called a mutagen.
It's a mutagen is anything that causes DNA to mutate. Okay, so there are chemical mutagens, but there are also environmental mutagens. So there are things that are just in your environment, like different kinds of light, that can cause your DNA to mutate. So when we talk about not going sunbathing, what kind of light is harmful for you? UV light, right? So UV light literally mutates your DNA, which is crazy. But that's why it can cause cancer, because the longer you're exposed to sun, the more mutations you tend to have, and then the more likely they don't get repaired, and that there are harmful mutations, so the mutation happens in the right place to then have you develop cancer. Okay? So yeah, there you go. That's why you're told don't sunbathe. Doesn't mean people don't still do it, but that's what the reasoning is behind that. So when we're talking about mutations, we're looking at the DNA being changed. So a mutation is any change to the DNA of a cell or even a virus. Viruses mutate as well. So when that changes, it means that the nucleotide sequence is different. So rather than AAT, you might have ATT or ACT or CAT. Any of those changes would be considered a mutation. Okay? So mutations tend to fall in kind of 2 different categories. There's the mutations that are listed here. So substitution, deletion, insertion. Those are different ways that the DNA can be changed. So you can insert an extra letter. You can switch out one letter for another. That would be substitution. Or you could have a letter deleted. You can also have more than one letter deleted. So you can delete 2 or insert 2. You could insert 3 or delete 3. But there's those 3 general kind of types of mutations. Based on which type you have, then you have different consequences or different results. So we can usually name mutations based on what happens to the code. Is something deleted, added, or just switched? So is it substituted? And then we can name it based on what that causes. Okay, so you can usually describe a mutation by at least 2 different terms. There's the class it falls into and then what it causes. So this half of the page tends to talk about the causes and why it's a problem. So sometimes you have a mutation that's called a silent mutation, nothing happens. So remember there's 64 different codes for 3 letters. So when we look at that circle chart to figure out what amino acid we have, some of them are duplicated. So you can have a silent mutation where you change one of the letters, but you still get the same amino acid. So the DNA's changed, but you still get the same protein, so it doesn't look like anything's different. You wouldn't even know that there's a mutation unless you sequence the DNA to see that it's changed, because your final result, the protein, is still the same. So that's called a silent mutation. You don't see it unless you're looking at the DNA. In a missense mutation, that's where you change one amino acid to a different one. So a missense mutation, where does that fit here? Substitution, deletion, or insertion? Substitution, because you're just switching one out for the other, okay? With a missense mutation, because of that change, that means that the amino acid changes as well. So in a missense mutation, you get a different amino acid. That can either affect the protein or not. So back when we talked about macromolecules and the fact that there are 20 different proteins, or 20 different amino acids, sorry, I talked about the fact that amino acids have different characteristics. So some of them are hydrophobic, some of them are hydrophilic, some of them are charged, some of them can form specific bonds with other types of amino acids. So if we have a hydrophobic amino acid and we change that for a hydrophilic amino acid, we have 2 things that have completely different characteristics, so the final protein is going to be different. That's what happens with sickle cell anemia. It was a missense mutation, so now we have a different amino acid, and changing one amino acid literally changes people's ability to carry oxygen around the body like they're supposed to, because their hemoglobin's now different. So sometimes it only takes one base pair, one thing being changed, to change the protein to completely change how the protein functions. You can have missense mutations that change the amino acid, but if you go from one hydrophilic amino acid to a different hydrophilic amino acid, it might not affect the protein at all. So it's a missense mutation that's a substitution, but it doesn't affect the organism. Okay. Another type of mutation is a nonsense mutation. So that's where you get a regular amino acid, but then it's changed to a stop codon. How many stop codons are there? 3. So you have a couple of different ways you could change a regular sequence to one of those. But if that happens, what happens to the whole protein? Say it, you're right.
Stops.
Yeah, translation stops. So if you put a stop codon in earlier than it's supposed to be, you never get a complete protein. So let's say the protein's 100 amino acids long. If we put a nonsense mutation in after 90 amino acids, then we mostly have the whole protein. We're only missing the end. So maybe it still works, maybe it doesn't. If we have a missense mutation happen after 10 amino acids, that's really short. You're not going to have a protein that works. So depending on where that mutation is, that determines whether you can still make the protein and it functions or not. Okay. A frame shift mutation is if you are inserting or deleting a base pair. So if you take something out or stick a couple of extra in, that triplet, so reading in triplet code, it has to shift. So if we started with these 3 here, and this is a base pair substitution. If we start with these 3 though, and we add 2 A's in the middle, we have GAA, and then the next triplet code starts with AA. So by adding or changing how the code is read, then you completely change all the amino acids that happen after that. So you're taking the reading frame, reading in sets of 3, and moving which direction it's read in. Okay? Does that make sense? Questions that I can answer for you?